Hello students, welcome you all in this video where I am solving model question paper 3 for the year 2025 and 26. Now I am going to solve 3 marks question from part C biology. Let's start with the question. So in this section, that is Roman number 14, answer the following question. We have to answer 3 questions. Each question carries 3 marks, so we are going to get 9 marks from this section. Question number 34 will start. Draw the diagram to show the structure of human brain and label the following parts. So here we have to draw the human brain diagram and we have to label hypothalamus and pons. So this is the diagram which I have taken from your NCERT book. Remember, you have to draw it very carefully, and after drawing this diagram you have to label here hypothalamus and pons. But in this diagram you can see I have taken all the labelings because this is just a practice what we are doing. So whenever we practice or whenever we solve any previous year question papers, you should practice with all the labelings. But in case whenever you are writing exams, then you have to label whatever parts has been asked. So here you can see this is called cerebrum. Here this is cranium or skull. Here this is a midbrain, spinal cord, pons, medulla and cerebellum all will conclude or include in the hindbrain. Then there is hypothalamus and pituitary gland. And this side here we can see that is forebrain. So brain is divided into 3 parts, that is forebrain, midbrain and hindbrain. So here in this question they have asked us to label hypothalamus and pons. So in this, if such type of question comes in exam, then remember you have to label this. Hypothalamus is here and pons is here. So these are the 2 labelings which you have to do if this question comes in exam. Otherwise, whenever you practice, practice with all the labeling, because not these 2, they may ask the another 2 also. So while practicing, practice with all the labeling. So this is the diagram of human brain. After this, we will move to the next question. Now the next is question number 35, which is divided into sub-question A and B. So we'll start with A first. Father only determines the sex of a child. Why? So as we know, in case of human being, in sexual reproduction, 2 individuals are required, father and mother. From father, male gametes will come, and from mother, female gametes will come, and both joins to get, after fertilization, will get a zygote, and that zygote will divide continuously and becomes an embryo. Embryo grows it and it becomes a fetus, and it takes 9 months, approximately 9 months, and after 9 months a new young one will take a birth. So in that case, only father can determine the sex of the child. This is the statement which is given and we have to tell why. So see the answer. Human have 23 pairs of chromosomes. So total pairs of chromosome is 23 in human being. Individually if you count, then there will be 46 chromosome. In terms of pair, it is 23. Out of this 23, one pair is called as sex chromosome. So remember, total there are 23. These 23 pairs of chromosomes are divided into 22 and 1. These 22 are the normal chromosomes. Remember, these are the normal chromosomes which are responsible for all the characters which are transferring from generation to generation, means parents to offspring. It may be like a skin color or height or eyes color, length of the hair, etc. Those are all characters. Normal characters will be transferred from father to mother. That all will be done with the help of this 22 chromosomes. But the one pair which is called as sex chromosome, that is responsible to decide the child will be boy or a girl. So see here, female has XX as a sex chromosome and male has XY as the sex chromosome. So female sex chromosome is denoted by XX, male sex chromosome is denoted by XY. Therefore, the mother always gives an X chromosome because mother has both XX. Whenever there will be fertilization, any one X comes, so always mother will give X chromosome for the fertilization. But if you see father has XY chromosome as a sex chromosome, so there are chances to give either X or Y chromosome during fertilization. So if the father gives X chromosome, then the child will be a girl child. And if the father gives Y chromosome, then the child will be a boy child. See here, very simple. I'll write it here, mother, and here I'll write father. So see here, mother has XX chromosome, father has XY chromosome. Here mother will give always X chromosome for fertilization, but father has the choice. He can give X chromosome or he can give the Y chromosome. If the sperm having X chromosomes combines with the egg, then the child will have XX as a sex chromosome and then the child will be girl. If the father gives Y chromosome and mother gives X chromosome, if it gets combined, the young one will get XY as a sex chromosome and the young one will be a boy. So see here, according to this all the explanation, as I told, mother has the capacity to give always X chromosome, then the sex of the young one is always depend on father because he has two different X chromosome, that is X and Y. So this is the reason for this question, that is father only determines the sex of the child. Question number 35. Now we will see the sub question, that is B from the same question. Now we'll see the sub question B. Yellow seed producing pea plant, that is denoted by capital Y, is crossed with green seed producing small y. Then draw a checkerboard showing the results obtained in F2 generation. So here they have given only one trait, that is color of the seed. You can see here we have two seeds, yellow seed. Sorry, we have two plants. One plant is giving yellow seeds and the another plant is giving green seeds. So only one trait is given. Therefore, this is a monohybrid cross. So I'll write it first. Answer, it is monohybrid cross. Monohybrid cross is very easy to write with the help of checkerboard. Monohybrid cross, I'll write it. What do you mean by mono? Mono means single. So here single trait is considered. For this trait we have to write F2 generation. So before writing F2 generation, first we have to write F1 generation. So we'll start with F1 generation. So I'll write down here F1 generation. So what we have yellow seeds. So I'll write here yellow seeds, and how it is denoted? Capital Y, capital Y. Then we have green seed, and here it is denoted by small y, small y. Now see here, capital Y means dominant. So these are dominant. And here small y means this is recessive. Now, capital Y will get a gene as capital Y. Here we will get a gene as small y. And in F1 generation, when we cross this, here there will be cross pollination. So this is cross pollination. Why this is cross pollination? Because both the plants are different. This first plant is giving yellow seeds, second plant is giving green seeds. So both the plants are different, and such type of pollination is called as cross pollination. So here we are going to get capital Y and small y. These are the genes we are going to get in the F1 generation. So I'll write it here. F1 generation, we are going to get all the pea plants with yellow colored seeds. Why yellow color? Because Y is capital. If there is a capital Y, then capital Y trait will be dominant. That is, yellow color of the seed will be dominant, and therefore all the plants we are going to get with yellow color seeds. Yellow seeds we are going to get in F1 generation. But remember, here one Y is capital, one Y is small. So these are called heterozygous. Heterozygous. And these both the plants were homozygous. Homo means same, pure plants. Because these both the plants are pure, these are homozygous. Here this is a hybrid, so it is heterozygous. Now by using this plant from F1 generation, we are going to do F2 generation. So now here I'll write F2 generation. Now in this F2 generation, what you have to do? We have to take the plant from F1 generation. So I will take the plant here. First one, what is that? Yellow seed, but yellow seed. But this is giving what? Capital Y and small y. This is heterozygous. Then in the same plant, we are going to do the pollination. So it is a self-pollination. So now let me write it separately here to show it clearly. So yellow seeds, and this will be again capital Y, small y. Here we are going to get genes, that is separately capital Y, small y. Here also we are going to get capital Y and small y. Now in these two plants we are going to cross it. Here we are going to do this crossing in the same plant which is getting from F1 generation. So it is called as self-pollination. And this crossing, this self-pollination or this crossing between these genes, we have to do with the help of checkerboard, because this is asked in the question. So let me draw the checkerboard on the next page. So here I have wrote everything once again so that we should not get confused again. Now let me draw the checkerboard. Now this is the checkerboard. Right of the genes, here I'll write capital Y, small y, and here I'll write same capital Y and small y. Now once this capital Y and capital Y get combined, here we are going to get capital Y genes in the young one. So we can write it capital Y, capital Y. Now see, both are capital. So this is dominant is yellow. So here we are going to get all the seeds of yellow color. Yellow seeds. Now if you see the next one, capital Y and this is small y, so we will get like this. But still there is one Y capital, so here also we are going to get yellow seeds. But remember, this is heterozygous and the first one is homozygous. If you see the next one, this and this, we are going to get capital Y and small y again. So capital Y, small y. This is again yellow seeds, but this is again heterozygous. It is not pure one. And the next one which is remaining is small y and small y. Now see, here there is no capital Y. Both are small. So here we can write, we are going to get green seed. This is homozygous, but it is green seed. Now, if you see here, what we are going to get is capital Y, we have 1. Capital Y, we have 2. Small y and small y, we have 1. So here genotypic ratio, if you see, it is 1:2:1. So this is the ratio. And if you see physical property, that is called as phenotypic ratio, how it is look physically, then if you write, I can write it here, yellow seeds. Let me write it properly. Spelling yellow seeds. How many plants of yellow seeds? 1, 2, 3. So we have 3 plants. And then the next plant is green seeds. Green seeds. How many plants are giving green seeds now here? See, this is the one. So I'll write it here 1. So the ratio of phenotypic is 3:1. So I hope you understood. Genotypic ratio, where we are going to see the gene. Phenotypic ratio, we are going to see the physical character. So here 1:2:1 is the genotypic ratio and 3:1 is the phenotypic ratio. So this is the answer for this question. Now we will move with the next one. Now the next is OR option for 35 question, that is red flowering tall pea plant. Now you can see here we have red flowering, flower color is red and a plant is tall. So we have two character. One is color and the second one is height. Whenever there will be two character, such type of crossing is called dihybrid cross. So tall is denoted by capital T, red is denoted by capital R, and this is a homozygous. homozygous because both are capital. Pure plant it is. Tall and red color both are dominant. Is hybridized with white and short. White and short. Short means small t. White means smaller. This is also homozygous. See here, this is also homozygous because both are small. Homozygous, but it is not dominant. It is recessive. Always small alphabet is used for recessive and capital alphabet is used for dominant. Remember this. This is very important. Now, then draw a checkerboard showing the results of obtaining F2 generation. So here also we have to show the F2 generation result with the help of checkerboard. And in case of dihybrid cross, it will be little bit difficult to show the F2 generation. So always we have to use checkerboard. So write down the answer. And you know that before showing F2 generation, we have to show F1 generation. So we'll start with F1 generation. Now take both the plants. So what is the first plant? Red flowering tall plant. So red flower and tall plant. Second one is white flowering short pea plant. So I'll write it here. Sorry. First we will write the color. White flower and short plant. See here. So I wrote here. Now we'll denote it. What is the see here? Capital R, capital T, capital T. Let me write it reverse because I have written here first red. So I'll write color first and then I'll write the height. So for this, I will write R, R, T and T. So see this. R, R means red flower. T, T means tall plant. Here it is small r, small t. Now what are the genes we are going to get from capital R? Capital we will write 1R. Capital T we will write 1T. Here capital small r we will write 1 small r. Small t we will write 1 small t. And once we cross this, we are going to get F1 generation. See this, both the plants are different. Whenever two different plants get crossed, it is called as cross pollination. It is called as cross pollination. And now we can write it here, that is capital R, capital T, small t. And what is this? You can see in case of color there is one capital R, in case of height there is one capital C. So whatever plant we are going to get in this F1 generation, all will show red flower and tall plant. Why tall? Because there is one capital T. Why red? Because there is one capital R. And you can see here, this is heterozygous. Heterozygous because there is capital and small mix, so it is heterozygous. Here we can say that in F1 generation, all the plants will be heterozygous with red flower and tall pea plant. Using this plants, we are going to do now F2 generation. So let me write that F2 generation on the next page. So write down F2 generation, and what are the plants we are going to use? The plant from F1 generation, that is nothing but red flower, tall plant. Both should be same. Or you can say we are going to do the pollination in the same plant. So therefore I'll write it here, R, small r, capital T, small t. R, small r, capital T, small t. Now if you see the genes, what we are going to get it here, how to write this? So you can do like this, you write R, capital T and capital T. Once you combine this R with capital T and then you combine with small t, then what you're going to get it here, capital R, capital T, capital R, small t. Then you combine this small r with capital T and then with this. So what again we are going to get? Small r, capital T, small r, small t. So these are the genes which we are going to get. So the same thing we are going to write it here. So write down the 4 for this and same 4 you have to write down for this because the plant is same. So capital R, capital T, capital R, small t, small r, capital T, small r, small t. See here, same thing I am writing. And now we will use this in checkerboard and we'll see the result in F2 generation. So let me draw the checkerboard. So write down the genes here. What you're going to get it? Capital R, capital T, capital R, small t, small r, capital T, small r, small t. Same thing. We will repeat this side also because we are going to do self-pollination now. In case of first F1 generation, there will be cross-pollination, but here it will be self-pollination. Remember, now it is self-pollination. Now combine and write down the result. Now see, if this and this get combined, whatever result you have to write it here. Then this and this you have to combine, then this, and then you have to do this. And same, repeat the procedure for whole this checkerboard. Now I'll write the first one. Capital R, capital R, capital T, capital T. See here, capital R, but capital T and small t. Capital R, small r, capital T, capital T. Then again capital R, small r, capital R, small r, capital T, small t. And like this, we have to complete this whole checkerboard. While writing this, you have to be very careful. Now I have a practice, so I am writing fast, but whenever you write it in exam or whenever you practice it, use pencil and write it very carefully, because if you go wrong here, then phenotypic and genotypic ratio both will go wrong. Therefore, you have to be very much careful while writing this checkerboard, and in case of a dihybrid class especially, because in monohybrid class it will be very easy. It will be not of that much confused, but in dihybrid class there will be more boxes to fill in this checkerboard, so you have to be very careful. So here I have completed this checkerboard. Now by seeing the dominant and recessive gene, you have to write the physical character. Like if you see here in the first box, if you see capital R, capital R, capital T, capital means both are dominant. So here we are going to get red color and tall. See here, like this we have to complete. Here also it is red, tall. Next also it is red, tall. Wherever you see R capital, it is red. If T capital, it is tall. So this is also red, tall. So see, like this you have to complete. First one is homozygous, remaining all are heterozygous, but still they are red, tall. Then the next is again red, tall. This is red, but it is not tall. You can see both the T are small, so it is short. Remember, this is again red, tall. This is red, but not tall. It is short because both the T's are small, recessive. This is red, tall. Can you tell me, can you write it by yourself? Next one, red, tall. Here both the R's are small, so it is not red, it is white, but it is tall. This is also white, but still it is tall because T is capital. I hope you are understanding. This is red and tall. Here it is red but not tall. It is short. The next is white, but still capital T we have, so it is tall. And the last one, if you see, it is white and as well as short. Like this we have to write the physical character. Now we will move with phenotypic ratio. So if you see now phenotypic ratio for this physical character, if you see now red, tall, how many we have? I'll write it here first, red and tall. Now we will calculate red and tall. I'll use the different color. I'll use blue color. Wherever there is red tall, I will mark it. So red tall 1, 2, 3, 4, 5, 6, 7, 8, and one more is here, 9. So red tall we have 9. I hope you understood now. Now the next one we will see red short. Now where are red short? I will use black color. Red short is here 1, Then red short is here 2 and red short is here 3. So I'll write it here 3. Now what is the next? White tall. So here I'll write white and tall. Now how many we have white tall? I'll use red color itself. 1, 2 and 3. So we have 3. And the next one, that is white and short. And white short we have only one, which is here. Now I'll write it 1. So you are leaving blue color. This is 1. So the ratio is 9:3:3:1. We have 9 red tall plant. We have 3 red short plant. We have 3 white tall plant, and we have 1 white short plant. So the phenotypic ratio is 9:3:3:1. In case of this, genotypic ratio will be not asked much in exam. That's why it is not given in the NCERT book also, because it will be a little bit confusing if you see this whole table. In case of monohybrid cross, we can write it, but in dihybrid cross, it is very difficult. So if you write only phenotypic ratio for such type of question, then also you will get the full marks. So here we have completed the answer of this question. I hope you have understood this answer. I have explained very clearly and neatly. Now remember one thing, in this they have asked red and tall. If they use any other characters, then also you should be able to solve this checkerboard. So for that you have to practice it. So after understanding this question, you use some other traits and try to practice it by your own in home. After this, we are going to move to the next question. If you are understanding this all the explanation of in the videos, if you are getting help of these all the videos, please consider subscribing my channel and share my videos with your friends. Now we'll move to the next question. Now the next is question number 36. A, because it has a sub question, grass, then the arrow frog, then the arrow snake. So it is a food chain which is given. Grass is eaten by frog, frog is eaten by snake. If the amount of energy in the first trophic level of this food chain is 100 joules, so the energy present in the first trophic level, that is in grass, it is 100 joules. What is the amount of energy available for the organism at the third trophic level? So here we have to use the 10% law of transfer of energy. Whenever energy is transferred from one trophic level to second, only 10% of the energy is transferred. By using that law, we have to answer this question. So here in answer, this is based on the 10% law of energy transfer. What the law says? Which says only 10% of energy is transferred from one trophic level to the next trophic level in a food chain. So according to question, here we have grass. So grass is coming under first trophic level, that is producer, which is going to produce the food. Frog is the second trophic level, that is called as primary consumer, means it is going to feed on this producer. Snake is the third trophic level, that is secondary consumer, it is going to feed on primary consumer. So whatever energy is present in the producer, out of that 10% will transfer into primary consumer. Out of this 10% will be transferred into secondary consumer. Only 10% is going to transfer, remember, and remaining 90% will be lost into the atmosphere. So therefore, in the question they have given energy at first trophic level, that is grass, we have 100 joule. Then we have to find out 10% is transferred in the second trophic level means how much. So see here, 100% means how much we have? 100 joule. Then how much will be transferred? Only 10% will be transferred. So 10% means how much? So we have to do here cross multiplication. If you do this, we are going to get, that is 10 into 100 joule divided by 100%. So see, 10 into 100 joule divided by 100%. Percent will get cancelled. And then what is remaining? So here 100 ones are 100, 100 ones are 100. What is remaining? 10. So the 10 and the unit remaining is joule. Therefore, only 10 joule of energy out of 100 only 10 joule of energy is transferred to the second, that is in the frog. Now, when the snake eats that frog, out of this 10, only 10% will be transferred. So, see here, energy at third trophic level. Now, we have only 10 joules. So, start 10 joule is nothing but 100%. So, 100% is how much now? 10 joule. That is in the frog. Out of this, only 10% will be transferred. 10% means how much? Now, again, we have to cross multiply. So, what will happen? 10% will go up into 10 joule and divided by it will be 100%. Now see what happens. 100% will get cancelled, and then what here? 10 ones are 10, 10 tens are 100, 10 ones are 10, 10 ones are 10. So remaining the answer is 1 joule. So that's why the answer is 1 joule. So from frog to snake, only 1% of energy will be transferred. So like this, if there is any another trophic level, then from this 1 joule, only 10% will be transferred. So same formula you have to go on using. So the energy available for third trophic level, that is in the snake, is 1 joule. This is the answer. Remember the method. If they change this number, that is 100 joule in the question, if in your final exam, use the same method and you can solve it for any number. So I hope you understood this answer. After this, we'll move with the next question. Now the next sub question, that is B. If all the waste we generate are biodegradable, what kind of the effects this would impact on our environment? Discuss. So whatever waste are produced in our day-to-day life or in our industrial life, etc., whatever waste is produced, if imagine all are biodegradable. What is biodegradable? Bio means living, degrade means decompose. If the waste is decomposed by using biological animals, biological organisms, living organisms, like it may be bacteria, it may be fungus, it may be virus, etc. If these all the living organisms are helping to decompose the waste, then that waste is called as biodegradable. And if it is not, then it is called as non-biodegradable, like plastic. Plastic cannot be decomposed by using this all the organisms. So it is called as non-biodegradable. But here in the question they have asked all the waste is biodegradable. Then what kind of effect you can see in the environment? See here answer. If all the waste are biodegradable, they would be broken down naturally by microorganisms like bacteria and fungi. All the waste will be decomposed by this bacteria and fungi. And this would have mostly positive effect on the environment, because if all the waste is eaten, decomposed by these bacteria, fungi, then there will be no waste into the environment which remains as it is for many days. Everything will get decomposed and therefore it shows positive effect. Which are the positive effects? I have listed here some of the effects. You can see the first one. Waste would decompose naturally and would not accumulate into the environment like plastic. We have seen the plastic. If you throw plastic into the environment, it will remain as it is for many years, because plastic cannot be broken down by these all the microorganisms. And if it repeats, then you can see the plastic waste everywhere in the environment, and that is called as plastic pollution. So that will avoided by using this method. Second, decomposed waste turns into manure or humus, enriching the soil. So whatever waste is decomposed by this bacteria, fungus, etc., all will be converted into manure and humus. And if all waste is biodegradable, then imagine the soil. Soil will be enriched a lot. The soil fertility will increase. That is the positive effect. Third one, animals would not suffer from ingesting non-biodegradable material like plastic. Nowadays we can see everywhere there will be waste, plastic will be thrown in the environment. If there is any biodegradable waste, for example, if there is a vegetable or unwanted food materials, waste, leafy materials, etc., all will be packed into the plastic and they will throw on the roadside. These all the animals like cows, buffaloes, what these all will do, they will go and try to eat that waste which is present into the plastic inside. And while eating this waste inside, they will eat this plastic also. And once they eat this plastic, it will remain as it is in their stomach, and it causes digestion problem in these animals. For example, if you see in case of dogs also, if you throw any food by packing into the plastic, dogs will try to eat that food, and while eating that food they will eat some amount of plastic also, and this also causes the digestion problem in dogs. So these all will be avoided if the waste present in the environment all will become biodegradable waste. So that is the meaning of this third point. So I hope you understood the answer of this question. So here I have completed the 3 marks question from this model question paper. I have explained very neatly in all the with the all points in the answers. Try to understand the answer. Make these all the notes in your book so it will be easy for you people to answer or prepare for your final exam. So if you like my videos, share my videos with your friends, and if you're getting little bit also help from this all the videos, then please consider subscribing my channel. Thank you so much.